Microprocessor complex subunit DGC8. The microprocessor complex subunit DGC8, DiGeorge syndrome critical region 8, is a protein that in humans is encoded by the DGC8 gene. In other animals, particularly the common model organisms Drosophila melanogaster and Caenorhabditis elegans, the protein is known as Pasha partner of Drosha. It is a required component of the RNA interference pathway. Function. The subunit DGC8 is localized to the cell nucleus and is required for microRNA processing. It binds to the other subunit Drosha, an RNase Roman 3 enzyme, to form the microprocessor complex that cleaves a primary transcript known as primiRNA to a characteristic stem loop structure known as a premiRNA, which is then further processed to miRNA fragments by the enzyme dicer. DGC8 contains an RNA binding domain and is thought to bind primiRNA to stabilize it for processing by Drosha. DGC8 is also required for some types of DNA repair, removal of UV-induced DNA photoproducts. During transcription-coupled nucleotide excision repair TCNER depends on J&K phosphorylation of DGC8 on serine 153. While DGC8 is known to function in microRNA biogenesis, this activity is not required for DGC8-dependent removal of UV-induced photoproduct. Nucleotide excision repair is also needed for repair of oxidative DNA damage due to hydrogen peroxide H2O, and DGC8-depleted cells are sensitive to H2O.